Hello everybody, you are welcome. In this video we will revise together paper 1 on cell structure part 1. Let's start. Number 1. A student used a light microscope to observe a blood smear on a microscope slide like this one, but not the same. An eyepiece graticule was used to measure the diameter of a white blood cell on the slide. The student recorded that the white blood cell was 5 eyepiece graticule units in diameter. Which additional information does the student need to determine the diameter of the white blood cell in micrometers? To allow the students to be allowed to measure the actual diameter of white blood cell in micrometers, he needs to calibrate the eyepiece graticule firstly. So, to convert eyepiece graticule units into micrometers, calibration with stage micrometer like this one is required. He aligned both eyepiece graticule as the base of eyepiece lens with stage micrometer slide on the stage of microscope and allow them viewed in the same field of view, and then count how many stage micrometer divisions equal or match with one eyepiece graticule unit. Okay, so we need only stage micrometer for this work. Letter E, calibration of the eyepiece graticule using stage micrometer only is the answer. Okay, while letter B, C and D are incorrect. Why? Because magnification of the eyepiece lens and magnification of the objective lens are not involved during calibration of eyepiece graticule scale. It's okay. Finally, you know also something about eyepiece graticule units or scale comes in divisions but without units, while stage micrometer come in divisions with known or definite units, okay, like micrometers for example. Okay, the answer is A. Number two. Which statement explain why it is necessary to use an electron microscope to see the chemistry of mitochondrion? Here in this figure, one mitochondrion seen by electron microscope, where chemistry and the folding of inner membrane are seen. Mitochondrion enveloped by double membrane and surrounded by a lot of extensive network of rough endoplasmic reticulum with tiny ribosomes on it. Here, which statement explain why it is necessary to use an electron microscope to see the chemistry of mitochondrion? This is a good chance to revise something about light and the electron microscope. Look please, for light microscope we use a visible light as source of illumination. Visible light with wavelengths range between 400 nanometer up to 700 nanometer. Okay, while electron microscope uses beams of electrons. Each electron has wavelengths equal or approximately about 1 nanometer only. Okay, so what's the relationship between the wavelengths used in microscope and the resolution of the microscope? What's the relationship? Resolution equal half wavelengths for light microscope. Look please, we used the minimum value of wavelengths 400 nanometer, so equal half times 400 equal 200 nanometer. This is the resolution power of light microscope. Here, light microscope can distinguish between 2 points if the distance between these 2 points is equal or greater than 200 nanometer. It can distinguish them as 2 separate points, while for electron microscope, resolution equal half times 1 nanometer equal half nanometer. It is able to distinguish 2 points. The distance between them is equal 0.5 nanometer or more. So the resolution power of electron microscope is much higher than that of light microscope. It's okay. Now, return to choices. The magnification of electron microscope is greater than that of the light microscope. The statement is right, but it is not the reason why we need electron microscope to see the crystal of mitochondrion. We need resolution power, higher resolution power, because the crystal is formed of membrane. This membranous structure is too tiny, to be seen by light. Even if we magnify with the highest magnification power of light microscope, we cannot see the crystal clearly. Okay, we need something clear like seen by electron microscope. So the answer A is not the answer, is incorrect. For letter B, look, the membranes of the crystal are separated by distance greater than 200 nanometer. If that's true, the light microscope can see it clearly. So the statement is wrong. Okay. The maximum resolution of microscope using visible light is too low. That's the answer. So our comment here, light microscopes lack the resolution to distinguish crystal due to the wavelength of visible light is too low. Okay, resolution power is too low. The answer is C. Let's check letter D. The wavelength of an electron beam is longer than the wavelength of visible light. According to our discussion, the statement is wrong. Look at the writing here. What about the wavelength of light? 400 nanometers, starting from 400 nanometers, while that of electron beam equal or approximately equal 1 nanometer. It's okay. Number 3. Some stains can be used to identify cell structures in living cells. A dilute solution of one stain causes the whole cell to appear blue. The blue color rapidly disappears from most cell structures. Those cell structures that releases energy stay blue. Which type of cell structure is likely to stay blue? Think, guess, which cell structure that release energy? Which one? Sure, right, you are right. Mitochondrion. So endoplasmic reticulum is wrong, Golgi body is wrong, lysosome is wrong, mitochondrion. Why? Mitochondrion is the organelle responsible for carrying out aerobic respiration, where most of reactions takes place inside mitochondrion. Link reaction, Krebs cycle reactions, electron transport chain reactions takes place inside matrix of mitochondria for link and the Krebs cycle, and electron transport chain reactions takes place on crest of mitochondrion. Okay, so the answer is D. Mitochondria release energy during respiration, aerobic respiration, and retains the stain longer than other organelles. Number 4. When mucus is secreted from a goblet cell, this event take place. Number 1, addition of carbohydrate to protein. Number 2, fusion of vesicle with the cell surface membrane. Extracellular release of glycoprotein. Separation of vesicle from the Golgi body. What is the correct sequence in which these events take place? Firstly, mucus is glycoprotein made of carbohydrate part and protein. Which one is formed firstly? Protein. Then we add certain organelles, add carbohydrate part to this protein to form glycoprotein. Look please, here carbohydrate is added to protein. This number 1. So we have to start with 1. The answer either A or B. Now look at this ribosome. This one ribosome. On membrane of rough endoplasmic reticulum. So that's rough endoplasmic reticulum. Here called cisterni, the lumen of rough endoplasmic reticulum called cisterna or cisterni. Cisterna. Okay? Inside the cisterna of rough endoplasmic reticulum, polypeptide that is formed due to translation of mRNA by ribosome will fold and attach with carbohydrate part added, and the polypeptide with carbohydrate folding takes place firstly in rough endoplasmic reticulum. Then certain vesicles pinch off the rough endoplasmic reticulum to form something called transport vesicle. This move along cytoplasm to reach Golgi body, where further modification and folding takes place in Golgi apparatus. Look please to this animation several times. Look here, nucleus, okay, rough endoplasmic reticulum, on it a lot of ribosomes like this black dots. Look, polypeptide chain is formed. Okay, movement within cisternae of rough endoplasmic reticulum, folding and addition of carbohydrate part. Look please again. Okay, then release transport vesicle, then attach with Golgi, another folding, secretory vesicle attached to cell surface membrane, where exocytosis of glycoprotein takes place at the end of this journey. Look, glycoprotein is released in this step. Look, exocytosis. So the sequence 1, 4, 2, 3. 1, 4, 2, 3. So the answer is A. Okay. Now number 5. The diagram shows 4 biological features: 1 DNA, 2 capsid, 3 70S ribosomes, 4 RNA. Which biological features are present in typical prokaryotes? Here this bacteria. Look at the bacteria. It possesses what? Possesses circular DNA, okay, of sure DNA genes transcribed into mRNA and several RNA like tRNA and rRNA for protein synthesis of bacteria. So DNA is present. Also what? Also RNA. So 1 of 4 are present for bacteria as prokaryotes. What else? Ribosomes with the type 70S ribosomes. So 1, 3 and 4 are present. What about capsid? Capsid is not present as structure in bacteria. Okay, so here prokaryotes have DNA, circular DNA, 70S ribosomes and RNA, but lack a capsid. Capsid is a unique feature for viruses, where capsid is formed from units of protein called capsomers, okay, surrounding the viral genome. So capsid is protein code for viral genome. So the answer 1, 3 and 4. 1, 3 and 4. So the answer is C. It's okay? Is C. Number 6. Which row shows features that occur in dicotyledonous plant cells and also in typical bacterial cells? The key here, take right, found means found in both dicotyledonous plant cells and also in typical bacterial cells. Take wrong means not found in at least one of these 2 types of cells. Also this means take wrong if this feature is found in one and missed in the another one. Also if both devoid of this feature or this structure, we take wrong, okay, or cross. Here for typical bacterial cells, look, it has what? Circular DNA. circular double helix DNA. Also circular DNA and 70S ribosomes. So look at chloroplast also in dicot plant. It possesses circular DNA, okay, and 70S ribosomes like bacteria. So both have 70S ribosomes and circular DNA. So the answer is B. Okay, the answer is B. Now look at this. Both plants and bacteria have 70S ribosomes and circular DNA. Advanced plant, dicot plant or dicotyledonous plants have no centrioles. Okay, all animal cells possess centrioles except nerve cell, neurons, while for plant cells it devoid of this structure. Advanced plants devoid of this structure. It's okay, so the answer is B. Number 7. The diagram shows transverse section through blood capillary. This wall of endothelium, one cell sac with narrow lumen with diameter equal 7 micrometer. What is the magnification of the drawing? To calculate the magnification of this blood capillary, we need to measure the image size or the scale bar size, okay, by using a ruler. Look, measuring firstly the scale bar image size, okay, by a ruler. Here image size equal 35 millimeter or 3.5 centimeter, okay. Then we use the formula IM, okay, IM to calculate magnification. M equal I over A, image size over actual size. So M equal 35 millimeter times 1000 micrometer. Here micrometer U, okay, micrometer divide 7 micrometer with micrometer cancel. To find the final answer, M equal 5000 times greater than the actual figure or actual size. It's okay, the actual size. So the answer is D. For 8, which statement explains why lymphocytes with no nucleoli die? Why lymphocytes with no nucleoli die? The cells do not have centrioles and cannot divide. Letter B, the cells do not have mitochondria and cannot release energy. The cells do not have mRNA and cannot transcribe DNA. The cells do not have ribosomes and cannot synthesize protein. What the nucleoli or nucleolus? Look at nucleolus, okay. What is its importance? What is its role? It is responsible for production of ribosomes, where inside nucleolus, by genes of nucleolus, rRNA is formed mixed with several types of proteins to form structures called large and small subunits of ribosomes. Then these ribosomes exit from the nucleus to start its job on the rough endoplasmic reticulum or even free in cytoplasm. It's okay. So D is the answer. Okay, firstly here nucleoli produce rRNA for ribosomes, which are essential for protein synthesis. So the answer is D. What about ribosome? It is made from mix between rRNA and the protein, certain types of protein. Letter A, B, and C. are incorrect. For letter A, centrioles are not essential for all cell divisions. Okay, what's the clue of this statement? Plant cell devoid, advanced plant cell devoid of centrioles, although they are able to divide, they are able to produce spindle fibers, even they have no centrioles. So centrioles are not essential for all cell divisions. Okay. Now for letter B, mitochondria are important for energy needed for all process in cell activities, but not directly related to nucleoli in this question. Okay. Letter C, DNA transcription is not dependent on nucleoli. Transcription of DNA genes is not dependent on nucleoli. So the answer is D. Okay. Now, number 9. Density gradient centrifugation is used to separate cell structures by their relative density. Larger cell structures have greater density and sink further down the centrifuge tube. What is the correct order of the cell structure starting from the top of the centrifuge tube? Look, firstly, we have to start sequence from top of the centrifuge tube. This type or this technique called cell fractionation by using ultra centrifugation by certain device called centrifuge. The principle or the basic scientific idea of centrifuge is the density of particles found in centrifuge tube. Firstly, when we homogenize certain tissue with certain type of cells, and by using, say, mixer or blender or even mortar and pestle, we obtain homogeneous or homogeneous solution. This solution, this extract of tissue, we put this diluted with certain isotonic buffer to avoid damage of organelles under study or all organelles of the cell. Okay, this solution centrifuged with high speed for certain period of time in centrifuge. We obtain two phases, supernatant liquid phase and certain solid pellet rich in nuclei at the first. Why nuclei deposit firstly? Because each nucleus has the biggest or highest density gradient or relative gradient than others. So we obtain nuclei. Then supernatant is taken into another test tube and centrifuged again with certain period of time with high speed. What's the result? Another pellet deposited at the bottom and supernatant above it. Here the pellet containing what? Containing mitochondria or mitochondria and the chloroplast if we use plant tissue. If we use plant tissue, we obtain chloroplast firstly because chloroplast is bigger and with higher relative density than mitochondria. So chloroplast then mitochondria. When we take this supernatant and fuse for the third time, we obtain the third pellet, lysosomes and other vesicles like transport vesicle, like secretory vesicle, like lysosomes, all these vesicles with one membranous structure. Then when we take this supernatant and finally centrifuge with very high speed for long period of time, we obtain pellet containing rough endoplasmic reticulum and a lot of ribosomes. So when make order from top of the centrifuge tube to the bottom, we obtain in this order. Look, in this order. Ribosomes comes first, so we start with ribosomes. So the answer C or D. Then mitochondria, lysosomes not found in our question, so missed, no problem. Here, which one is second order mitochondria? Because it's smaller than chloroplast. Then we reach nuclei or nucleus. So the answer is D. Okay? Density gradient centrifugation separates by size, by density, relative density gradient. With ribosomes smallest at the bottom, at the top of test tube, and nuclei largest at the bottom. Answer is D. Number 10. Which cell structures contain nucleic acids? Sorry. Chloroplast, Golgi bodies, lysosomes, ribosomes. Look, chloroplast containing what? Circular DNA. So number 1 is right. What about chloroplast containing DNA and RNA? Both are nucleic acids. And ribosomes also containing rRNA. Okay, rRNA. What about Golgi bodies and lysosomes? Look, this wrong, this also wrong. So the answer is C. 1 and 4 together. Okay, or 1 and 4 only. Number 11. The statements describe processes that take place in secretory cell. Number 1, modification of the protein occurs in the Golgi body. Number 2, mRNA leaves the nucleus. Number 3, ribosomes bind to mRNA during translation. Number 4, transcription of specific DNA sequence occurs. Number 5, vesicles fuse with the cell surface membrane. Number 6, vesicles transport the protein to the Golgi body. Statement 4 is the first process, and statement 5 is the last process. What is the correct sequence of the 4 middle processes, or the middle 4 processes between 4 and 5? Look please to this figure. After transcription of the DNA, mRNA is formed. The result of transcription, mRNA. So mRNA leaves the nucleus, or ribosomes bind to mRNA during translation. So number 2 is first. The correct sequence follows protein synthesis. Look please to this figure again. After and during translation, look. Inside nucleus, DNA transcription. mRNA leave the nucleus as message to reach ribosomes, where translation takes place. So the correct sequence, mRNA number 2. Okay, leaves the nucleus. Ribosomes number 3. Ribosomes bind to this mRNA message during translation. Number 6, transport to Golgi. Okay, number 6, transport to Golgi, then exocytosis finally, but at Golgi, modification of protein as glycoprotein or glycation process takes place inside Golgi apparatus. So the sequence 2, 3, 6, 1. So the answer is B. Okay, is B. Number 12. How many types of structures with a double membrane that are found in animal cells are also found in plant cells? Look please to this figure. Organelles may be non-membranous or membranous. For membranous one, may be single membrane like lysosomes, endoplasmic reticulum, Golgi bodies. While with double membrane, it is called envelope, nuclear envelope, chloroplast envelope, mitochondrial envelope. It is formed of 2 membranes, inner membrane and outer. It's okay? Here, the question again. How many types of structures with a double membrane that are found in animal cells and are also found in plant cells? Concentrate on this. How many structures? Nucleus, chloroplast, mitochondria, 3. But here's a question about animal cells. Animal cells devoid of what? Devoid of chloroplast. No chloroplast found in animal cells. So with double membrane structure sharing between animal cells and also in plant cells, two structures. So the answer is B. Only two structures, so the answer is B. Okay. Number 13. A light microscope is used to observe two structures that are 200 nanometer apart on the slide. This actual distance between two structures found on the slide and seen by light microscope. What is the actual distance between the two structures when the magnification is changed from 40x to 400x? Sure, it is still the actual. It still keep the same distance. I mean keep the 200 nanometer. It doesn't affect with the magnification power of the lens used. The actual is not affected by the lens used or its magnification power. So the answer is letter C, still 200 nanometer. The actual distance between the two structures does not change when magnification changes. Okay? Since the original distance is 200 nanometer, it remains 200 nanometer regardless of the magnification used by eyepiece lenses or objective lenses. It's okay? So the answer is C. Number 14. A cell is shown in the micrograph. Which statement explain how it is possible to identify the type of microscope used to produce a micrograph? The nucleus is visible, so an electron microscope was used. Letter B, the endoplasmic reticulum is not visible, so a light microscope was used. Chloroplasts are visible, so a light microscope was used. Ribosomes are visible, so an electron microscope was used. Look please. This figure or micrograph is taken by using TEM, transmission electron micrograph. Okay, which statement explain? Letter E, the nucleus is visible under a light microscope also. Can be visible. Look at this figure where the nucleus is visible and even its nucleolus also visible by using light microscope in this figure. So letter E is not the answer. For letter B, the ER cisternae is not visible by LM. Okay, LM light microscope cannot visualize rough endoplasmic reticulum. Okay, so letter B is wrong. Letter C, chloroplasts can also be seen in both light and electron microscope. We can use light microscope to visualize chloroplasts inside plant cells. Look, this figure or this animation of sure taken by light microscope. As you know, light microscope, one of its advantage to visualize specimen and can make videos, animation. Okay, living tissue. Here living tissue visualized by light microscope with a lot of chloroplasts surrounding the vacuole at the middle or at the center of each plant cell. It's okay. So A, B, and C are wrong. Ribosomes are visible. Ribosomes are the tiniest organelles inside cells. Okay, with about 20 up to 25 or from 18 up to 25 nanometer according to the type of the cell. So the answer is D. It's okay. Number 15. Which statement supports the fact that mature plant cells contain organelles that carry out the same role as lysosomes? Letter E, range of hydrolytic enzymes can be found within mature plant vacuoles. Letter B, glycogen found within vesicles can be hydrolyzed to glucose molecules. Letter C, double membrane bound vesicles are formed from Golgi bodies. Letter G, vesicles formed from the cell surface membrane contain enzymes. Firstly, we deal with mature plant cells and lysosomes. Okay, and lysosomes. Lysosomes found in animal cells. All animal cells contain lysosomes. Lysosomes containing wide range of hydrolytic enzyme, proteases, carbohydrates, lipases, all of these enzymes found in lysosomes. Lysosomes are formed from Golgi bodies as single membranous structure. Okay, what is the similar structure or organelle inside mature plant cells that are doing the same role as lysosomes? Look, firstly, this vacuole, plant vacuole, large permanent vacuole, in most plant cells carry out what? Storage and support. Support as it maintains turgor pressure, okay, and also containing storage material like water, mineral in the form of ions, positive and negative ions, sugar, okay, soluble sugars, enzymes, waste products. Waste products inside vacuoles are broken down by what? By digestive enzymes, by hydrolytic enzymes. So in plant cells, vacuoles contain hydrolytic enzymes that break down waste materials. So the answer is A. Okay, what about B? Look at glycogen. Glycogen is polysaccharide found only in animal cells, stored carbohydrate for long-term storage material, in muscles, in liver, okay, found in animal cells, not found in plant kingdom or plant cells. The storage material inside plant cells is starch. Plants store starch, not glycogen. For letter C, Golgi vesicles help transport proteins, but do not replace lysosomes in its function. For letter D, these vesicles that formed from cell surface membrane for transportation material but not for digestion like lysosomes. So the answer is A. Number 16. Which cell structures may contain cisternae? Chloroplast, endoplasmic reticulum, Golgi body, mitochondrion. Take right, may contain cisternae. Take wrong, does not contain cisternae. What is meant by cisternae? Cisternae are membranous structure, membranous sacs, okay, found in certain organelles. Look, firstly, for chloroplast. Chloroplast is formed from outer and inner membranes as envelope. Thylakoid membranes in the form of grana or granum, okay, spread within stroma, okay, with certain structures. So no cisternae found in chloroplast. So the answer C or D. Okay, look here. Cisternae are flattened membranous sacs, okay, found in the endoplasmic reticulum, yes, present in endoplasmic reticulum like this figure, and the Golgi bodies. Here endoplasmic reticulum is folding containing cisternae or internal space called cisternae, okay, found in endoplasmic reticulum, rough and smooth, and also Golgi bodies, okay. Mitochondria and the chloroplast do not contain cisternae. Look at mitochondrion, formed from double membranes, outer and inner, and the matrix. Okay, the inner membrane called cresti, but no cisternae found in mitochondrion. So the answer is C. Okay, it's C. Take right in endoplasmic reticulum and take right in Golgi body. Number 17. This diagram shows some cell structures of one type of cell from 1 to 5. Which label structures, cell structures are present in typical eukaryotic cells and typical bacterial cells together? Number 1, cell surface membrane found in both. Okay. Number 2 is a capsule found in what? Found in prokaryotes. In some types of prokaryotes, some types of bacteria containing or surrounded by capsule for extra protection, but not found in eukaryotic cells. So 2 is wrong, is missed. Not sharing common features. What about DNA? Circular DNA or even DNA? DNA found in both eukaryotic cell and typical bacterial cells. Cytoplasm in both, ribosomes in both. So here with numbering, cell membrane number 1, DNA number 3, ribosome number 4, and cytoplasm number 5 are all common structure of all living cells. Now some bacteria have capsule, not all bacteria surrounded by capsule. Look at this figure. Here is a capsule or slime layer. This additional protection structure sometimes present in some species or some types of bacteria. Here structure always present, all these structures present in prokaryotes, plus additional features sometimes present in some types of bacteria. It's okay? Okay. The answer is B. Okay. Now the answer is B. 2 is missed and the answer is B. 1, 3, 4 and 5 and 2 is omitted from our answer. Okay. Number 18. Which steps are needed to find the actual width of a xylem vessel viewed in transverse section using a 10x objective lens? Here, xylem vessels found in vascular bundle and dicot stem. Look at this figure. It contains a lot of xylem strands with a lot of mini xylem strands with a lot of xylem vessels. Okay, you're beside me. Number 18. Which steps are needed to find the actual width of a xylem vessel viewed in transverse section using a 10x objective lens? Here, vascular bundle containing many xylem strands with a lot of xylem vessels, and overlay above it a scale. It's called eyepiece graticule. Okay, scale with many units. How to find the actual width of xylem vessel? We measure many xylem vessels because they are different in size and take the average mean with many diameters to take the average mean diameter of the xylem vessels. And number 1, convert from millimeter to micrometer by multiplying 10 to the power minus 3. When we need to convert from millimeter to micrometer, we have to multiply by 10 to the power 3. Okay, not this figure. It is wrong. Now for calibration. Calibration is needed by using stage micrometer, right? But on a 4 objective lens is wrong. Why? Because here the transverse section is taken by viewing or using 10x objective lens. So we have to calibrate at 10x, not 4. Okay? So the statement number 2 is wrong completely. We need to calibrate, but at what? At 10x objective lens. Number 3. Look at this figure. Here when make calibration of eyepiece graticule with stage micrometer at 100x as magnification power. 10 for eyepiece lens and 10 for objective. 10 times 10 equal 100. Look at the value of each eyepiece graticule unit and compare it with when we change the objective into 40 instead of 10. Look, now the magnification power equal 400. What is the value of each small division of IB's graticule? Becomes different. So statement number 2 in the question is wrong completely. We have to calibrate by using the same objective lens. Calibrate as the same objective lens. I mean by using 10x objective lens. It's okay. Now for summary of this slide. Look, to measure the actual width of the xylem vessel, measure the width using eyepiece graticule, how many eyepiece graticule units, then multiply by the number of eyepiece graticule units by the calibration value, how many micrometers. Using the value of eyepiece graticule unit differs as the objective lens used differs. It's okay. So the answer now, then calibrate the eyepiece on 10x objective lens as in question. So the answer now is 3 and 4. 3 and 4, so the answer is D. It's okay. The answer is D. Number 19. Which cell structure is found in human cells and typical plant cells? 70S ribosomes, cilia, plasmodesmata, tonoplast. Look, for both. Firstly, human cells, human and plant cells have 80S ribosomes, but mitochondria in both and the chloroplast in plant cells contain 70S ribosomes. For letter B, cilia. Cilia only present in some human cells but not in plant cells at all. Here, ciliated epithelial cells found in lining of trachea or bronchus. For letter C, plasmodesmata, found only in plant cells like this one, this gaps between neighboring cells, here, plasmodesmata, okay. For letter D, tonoplast, found only as membrane surrounding vacuole, present only in plant cells, like here, this tonoplast, okay, tonoplast. So the answer is A. 20. Which statements are correct for a typical prokaryotic cell? It contains 70S ribosomes. Yes. It contains cellulose cell wall. No. It contains circular DNA. Yes. It is up to 5 micrometer in diameter. Yes. So prokaryotic cells contain 70S ribosomes. Number 1. Bacteria have peptidoglycan instead of cellulose as cell wall. Look at peptidoglycan in both types of or groups of bacteria, gram-negative bacteria or gram-positive bacteria. Peptidoglycan found as cell surface membrane instead of cellulose. For letter circular DNA, yes, present and are up to 5 micrometer in diameter. Number 4, right. So 3 is missed, 2 is missed, sorry. 2 is missed in our answers, so the answer is C. Number 21, the diagram shows a typical animal cell. What's the function of the membrane system labeled X? X is considered cisterna of rough endoplasmic reticulum, where it is needed for protein modification and transport as certain vesicle bud of its surface. So here, rough endoplasmic reticulum is involved in protein synthesis where ribosomes are attached to its surface and also needed for transport via vesicles, so the answer is B. Now, for letter A. A is considered smooth endoplasmic reticulum. This for smooth endoplasmic reticulum. Lipid synthesis occurs in the smooth endoplasmic reticulum. It's okay, so the answer is B. 22. What describe a lysosome? A vesicle containing enzymes enclosed by a double membrane that is budded off the endoplasmic reticulum. Letter B, a vesicle containing hydrolytic enzymes and surrounded by a single membrane found only in phagocytes. Letter C, a vesicle enclosed by a single membrane containing several different hydrolytic enzymes that may act inside or outside the cell. Letter C, a vesicle surrounded by a double membrane containing enzymes which can hydrolyze damaged organelles in a cell. Look at this short video to show the function and role of lysosome and its structure also. Here two lysosomes containing hydrolytic enzymes. These enzymes firstly are synthesized by rough endoplasmic reticulum as they are formed from protein. Pinch off firstly as transport vesicle combined with Golgi apparatus, fused with Golgi, where extra modification takes place to form lysosomes. So lysosomes are formed directly from Golgi bodies. Here bacteria by endocytosis taken up by this phagocyte to form phagolysosome or phagosome firstly, then phagolysosome, where lysosomes break down the pathogens into functionless parts and antigens are formed, for example. What about the another function here? Look please again to this part. Another lysosome fuse with worn out organelles like senile mitochondrion and degrade its structure and recycle its components. It's okay. So now for lysosome again. Lysosomes, a single membranous structure, not a double, containing a lot of hydrolytic enzymes found in all animal cells, not only found in phagocytes. Look please. It has a lot of number in phagocytes, but found in all animal cells. Lysosomes contain hydrolytic enzymes for breaking down waste materials, okay, or even worn out organelles like mitochondrion or mitochondria. Letter B, only in phagocytes. This is wrong, where lysosomes exist in all animal cells. For letter A and D, double membrane, lysosomes are not double membrane, they are single membranous structure. Okay, so the answer is A. Okay, the answer is A. Sorry. So the answer is C. Okay, is C. Thank you for your watching. Thank you for watching. See you later in many other videos. Good luck. Best of my wishes. Bye bye.